And the first talk will be by Nicole Kleinstreuer. She has studied mathematics and biomedical engineering. She received her PhD in bioengineering and completed her postdoctoral training at the US EPA Center for Computational Toxicology. She is now acting director at the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods and NICETM. And at NICETM, she leads the computational toxicology work. Nicole Kleinstreuer has received several awards for her contribution in the field of toxicology, especially in the field of alternative toxicological methods and computational modeling. And we are looking forward to her talk.
Thank you very much for the kind introduction. I'm sad not to be there with you in person, but I'm very happy to be able to join you virtually to give the talk. I'm not at the US EPA. I used to be. It was a few years ago now. But as the intro indicated, I am with the NICETM, which is the US Federal Resource for Alternatives to Animal Testing. So please go on to the next slide. So I'm actually going to speak less today about the actual models themselves and more about the process of looking at the challenges and increasing the trust of both building and adopting models into regulatory use. Because I think a lot of people have heard me speak a lot about the computational models that we've built. And I think some of the really exciting developments are in the space of actually understanding how to tailor those models to regulatory needs and decision contexts and how to encourage their adoption and implementation. So just a tiny bit of background about NICETM and ICVAM. We were established over 20 years ago now by an act of the US Congress to bring together all the different US federal agencies that either require or consider chemical safety testing data and are interested in moving towards more rapid, more efficient, and potentially more human relevant approaches to testing chemicals. So you can see the agencies that we support on the bottom there. They are about evenly divided between regulatory agencies and research agencies, and we are also partners in the Tox21 project. Next slide, please. So this document was published almost 5 years ago now. So this is the U.S. Federal Agency Roadmap to establishing new approaches to evaluate the safety of chemicals and medical products. And this was really driven by sort of this quote at the top of the slide, which is that we've had these tremendous advances in science and technology, but by and large, they haven't really been adopted into regulatory use because there has to be a lot of consideration around how they might be used to address current regulatory needs and decision context. It's not enough to just come up with a cool technology and validate it and then give it to a regulator and say, here, please use this, if you haven't taken into account what the needs of the regulator are from the beginning. And so these three pillars really underpin the foundation of this roadmap, which is to involve the end users and the regulatory decision makers from the very beginning in kind of communicating their needs and helping to guide the development of new methods that might meet those needs, and then having efficient and flexible approaches to establish confidence or validate those new methods that ultimately will support the adoption of those methods. Next slide, please. And I think the really key learning that we took away when we were developing out this roadmap is that there is no such thing as a one-size-fits-all approach to establishing confidence in these new approaches. Next slide, please. So I think the sort of old sort of historical viewpoint on validation was that validation in and of itself was something that could just be achieved. So you have a new method and then you go to a validation body and they validate it, and then it's approved for regulatory use. Next slide, please. But the reality is very different. As I said, we support 17 different federal agencies, and this is just an example of two of those agencies, the US FDA and the US EPA. And even within those individual agencies, there are a number of different centers and offices that focus on different types of chemistries, different types of products, ranging from drugs to devices to food additives to pesticides to industrial chemicals. And all of them have their own unique decision context and their unique considerations that inform how the concept of validation has to be carried out to make sure that new approaches are actually going to fit their needs and fit their regulatory frameworks. So next slide, please. So in putting together the strategic roadmap, we came up with another sort of acronym. So equally as important as the 3 R's, so reduction, refinement, and replacement of animal testing, is the 3 C's. And this is communication, collaboration, and commitment. And only through these 3 C's have we been able to really make tremendous progress in adopting and implementing new approaches, including computational models. So for the next couple slides, next slide, please, I'm going to talk through how we are updating the guidance on validation and regulatory acceptance of new approach methodologies, and really doing that with an eye towards how to implement not only in vitro methods, but also computational models into regulatory use. So this is the old ICVAM guidance on validation. It was published in 1997. So we figured 25 years, that's probably a good time to revisit this guidance and give it at least an update, if not really a complete overhaul. And that's basically what we're doing. We have a number of different agencies extremely engaged, from the FDA to the EPA to the Veterans Affairs Administration to occupational safety and health agencies to the Consumer Product Safety Commission. And they've all been very, very active over the last several months in trying to write a new guidance on validation. Next slide, please. So what does that transition look like? Really, we're going from sort of this centralized one-size-fits-all type of model where you consider a method as either validated or not, and kind of think about them as standalone replacements, towards a much more decentralized model where our end users, predominantly our regulatory decision makers and stakeholders, are really helping to guide that process and ensure that the evolution of establishing confidence in a model is fit for the intended purpose, and it's not a static yes/no outcome. It is an iterative, sort of mutually informative process that gets you to a place where you can have integrated strategies that provide more or less confidence for particular applications. Next slide, please. Obviously, there are a number of existing guidances that we're using to inform this new document. OECD GD34 has a number of really fundamental principles in it that really do remain the same in this new guidance. We just sort of expand upon them a little bit more and try to give them more context. And we are introducing here this context of use terminology, which is essentially the same thing as the purpose of the method, but it takes into account really the regulatory application. The emphasis in the new guidance is really on flexible and adaptable processes to establish confidence in methods and ultimately validate them for regulatory use. And coming back to one of the C's and the 3 C's, having constant communication, not only between the end users and the method developers, but among different regulatory stakeholders and end users, because there are different needs across different federal agencies, not only within the US, but globally. So next slide, please. This is a preview of the kind of main figure of the document on the key concepts of flexible, fit-for-purpose validation of new approaches. And those key concepts are ensuring biological relevance of the system, integrity of the data and transparency of the information, and adequate technical characterization, such that an independent body can come in and really look at all of the information that's being provided by the method with respect to its context of use and qualify or validate or establish confidence in that approach for regulatory decision making. Next slide, please. So some of the details of what we have in this new guidance on how we're trying to kind of create these more efficient, robust, and flexible practices to validate new methods is, again, to start from what are the testing requirements that this method is intended to fulfill and the context of use. And have communication with not only our regulatory decision makers, but with the public and the scientific community to understand where the science is and how it can best be adapted and applied to fulfill those regulatory needs. And always doing this with an eye towards global harmonization. So I think some of you are aware, and some folks from the BFR are actually involved in a OECD initiative to try and update the test guidelines program process to evolve how validation is considered and applied within the context of new approaches, in vitro methods, in silico computational models, et cetera, considering things like validating individual information sources versus a full defined approach, et cetera. And so there's going to be a workshop at the beginning of December for the national coordinators of the test guidelines program to discuss these important considerations and how the OECD can also evolve to keep pace with a need for flexible approaches to validation that are fit for purpose. Next slide, please. Some of the detailed sections within this new validation guidance are looking at how you can define relevance of new approaches. So the biological plausibility and the physiological process that's being measured in an assay or is being modeled by a computational model, and how that relates to mechanistic relevance for a human health endpoint or an ecotoxicological endpoint that you're trying to predict. Looking at reference data and how you really get to that standard of new approaches must be as good as or better than the existing reference animal data, which is generally legacy in vivo studies. So how can you examine that reference data and really characterize it in a very realistic way? Look at the variability or the uncertainty in that sort of gold standard reference animal data and use that to inform how you evaluate the performance and the applicability of new approaches that are intended to potentially replace that existing legacy test. And then a lot of information on technical considerations and data quality tools and best practices for quality systems development. And I have at the bottom of the slide here two very recent papers that have a lot of these topics that are going to be considered in the new ICVAM validation guidance. The van der Zalm et al. Paper, it looks at a scientific confidence framework, and the Peterson et al. Paper looks at a technical framework for high quality new approach methodologies. Next slide, please. So our role here is, ICVAM's role is really to kind of assure an independent process for establishing confidence in these new approaches, to ensure that we have communication and collaboration among different federal agencies, again, domestically and internationally, so that we can ensure that we are taking an approach that is harmonized globally and meets the needs of regulatory stakeholders worldwide, not just in the US. Next slide, please. And so where are we with this document? We are in sort of the final drafting stages through the validation work group, and then it will go through clearance at 17 federal agencies, which, as you can imagine, is not the most straightforward process, but we're hoping that it'll go relatively quickly. And then we're going to send it to our stakeholders, which includes our international partners, like some of our collaborators at the BFR and at the OECD and other regulatory organizations worldwide, and give them the opportunity to comment on the document before we put it out for public comment. Okay, next slide, please. So now I'm going to talk through a couple of different examples where I think that these concepts in terms of flexible fit for purpose validation are already being applied from a computational modeling and an in vitro assay perspective. So this is an example from the multi-agency collaboration called APCRA, or Accelerating the Pace of Chemical Risk Assessment. So this is a collaboration between us, the National Toxicology Program, ECHA, the US EPA, Health Canada, ECFAM, the European Commission, DG Environment, and several other partners. And this is a paper that was led by Katie Paul Friedman that was published in 2020. And so we basically took points of departure based on high throughput screening, so data from the Tox21 and ToxCast programs, looking at bioactivity in a range of different in vitro assays, and then applied mathematical modeling, so in vitro to in vivo extrapolation, to derive these POD NAMs, so point of departure, that would be essentially the amount you would have to be exposed to externally to achieve internal concentrations where we were seeing activity in the in vitro assays, and then compared those with exposure predictions and with points of departure based on traditional animal studies for about 450 chemicals. And the interesting thing here is that for about 90% of the chemicals, the point of departure based on the new approach methodologies, based on the in vitro data, was conservative, meaning that it was, less than the traditional point of departure, but not quite as conservative as taking sort of a TTC approach. And then for the other 48 substances or so, the PODs were very close to one another. So they were typically within a factor of 10. And there was some structural enrichment there within particular chemical classes like organophosphate and carbamate insecticides, which kind of gives us some insight into particular structural categories that we may need to look a little bit deeper into, better representing their mechanisms of toxicity with in vitro assays or potentially in silico structure-based approaches. And so I think this is a really nice demonstration of a fit-for-purpose approach to screening of chemicals. So here, it's feasible that you can use in vitro bioactivity as a protective estimate of the point of departure in screening level assessments. And there's currently work ongoing within APCRA to look at more of a prospective case study of an additional 200 substances. Next slide, please. The next example I want to talk through is an example of using the variability in the reference data to provide context to the predictions that are derived from a QSAR model. So here we took a sort of a global crowdsourcing collaborative approach to build a consensus model for acute oral systemic toxicity. So we've done this with a number of different endpoints like endocrine disruption in collaboration with the EPA and Prachi Pradeep, who used to be at the EPA, is now at the BFR with you all. You're very lucky to have her. And the most recent version of this kind of collaborative approach to model building was to predict the oral LD50 from rodent studies. And so we had 35 different groups around the world contribute models to predict things like the GHS categories or the EPA hazard categories, or delineate very toxic substances from non-toxic substances, or actually quantitatively predict the LD50. And we got over 130 different models submitted to us. And then we used an external test set to qualitatively and quantitatively evaluate the robustness and the performance of those models, and then combine them together into a consensus modeling suite that we call CATMOS, or the Collaborative Acute Toxicity Modeling Suite. It is contained in OPERA, which is an open source kind of toolbox of QSAR models. It's actually also now included in the latest release of the OECD toolbox as a plugin. And here, to really evaluate the performance of this consensus QSAR model, we relied upon the in vivo reproducibility of the rodent oral LD50 test. So we had several thousand chemicals where we had independently replicated acute oral LD50 data. And so we were able to use that to derive a 95% confidence interval by bootstrapping through the standard deviations from the replicate tests. We found that basically a quarter log in either direction, so plus or minus 0.25 log 10 mg per kg per day, represents the inherent variability in the animal data. And so if that's how well the animal data are able to predict themselves or reproduce themselves, then that sets an appropriate threshold for expectation of model performance when you have a computational model that's attempting to predict that endpoint. And so the variability analysis was published just a couple months ago in ToxSci, and then the assessment of the OPERA CATMOS model was published in EHP, and you can see that the QSAR predictions perform just as well as the in vivo data do in reproducing themselves when you're looking at predicting the acute oral toxicity output. And this model right now is actually being evaluated by the US EPA in a large retrospective analysis where they're looking at the past 50 or 60 years of risk assessments to see if you substituted the predicted LD50 value from the model for the measured LD50 value that was used as the basis of the risk assessment, what would the outcomes be? How often would you miss a case of suspected risk, or how often would you be more conservative? And that work is ongoing. We're hoping to submit it for publication ideally by the end of the year, but sort of a spoiler alert, it does really well. And we're hoping that it will actually form the basis for regulatory guidance, particularly for non-toxic substances, because as it turns out, if the model predicts an LD50 above 2,000 mg per kg per day in over 90% of cases, that's very consistent with the in vivo data. And in a much higher percentage of cases, almost 100% of cases, it results in the same risk assessment outcome. So that's pretty exciting. Next slide, please. The next two examples are more in vitro relevant examples, but this is a paper I wanted to bring your attention to. It was published last year, and it was actually awarded the Paper of the Year from the Alternative Specialty section at the Society of Toxicology annual meeting. And it basically takes a new approach to establishing confidence in methodologies for assessing eye corrosion and irritation potential, because an analysis of the existing animal reference standard shows that Drai's rabbit eye test is very unreliable. It's irreproducible, particularly in those middle categories. There's mild and moderate irritants. And so anytime that you build a model that tries to predict an endpoint that is in and of itself so variable and so irreproducible, then you're introducing all kinds of noise into your model when training that system. And so if we take a different approach and we just look at all the available methods and understand how do they actually represent human ocular anatomy and physiology, and assess all of those different models, both in vitro and in vivo, in terms of their ability to look at things like the depth of injury in the eye, then it's a very different approach and can be used to validate either individual in vitro methods or combinations of in vitro methods in defined approaches. Next slide, please. And then the last example that I'll just mention very briefly that everybody's probably very familiar with is how effective it can be to come up with defined approaches that are anchored in human biology. So here we have the adverse outcome pathway for skin sensitization, and several defined approaches were included in this recently published OECD guideline, including for the first time in silico or computational information sources forming parts of defined approaches. And here we could compare to the animal reference standard and show that the defined approaches were as good as the LLNA. But in this case, we also had the luxury of comparing to human data, largely due to the tremendous efforts by Matthias Hertzler and other BFR colleagues in helping to curate high quality human reference data. And here we see that human biology-based defined approaches actually outperform the animal data when compared to human reference data. Next slide, please. So we've learned a number of lessons along the way in kind of building out this roadmap and trying to implement it and trying to update the validation guidance and trying to apply these flexible, fit-for-purpose approaches to validating both computational models and other new approach methodologies for specific regulatory needs and decision contexts. So I won't read those to you. You can see them yourself, but I will just say that this is a continuous process and we're constantly learning and adapting, and we're very grateful for our international colleagues, particularly those of you from the BFR who have been really engaged in a lot of these projects. So last slide is just acknowledging my amazing group that I'm very privileged to lead, and just drawing your attention to a couple of online resources like our integrated chemical environment, the ICVAM biennial report, which talks about all of the different activities and alternatives at different US federal agencies, and our newsletter, which comes out about weekly. Okay, thanks so much.